Hello, let's continue with our Unit 6 notes. We are on slide 19, so make sure that you, well obviously you must be because you did your bell work already. And that should have been pretty simple. We've been doing that 2 or 3 times already for different activities, different assignments in the class this year. So let's jump into the unit. This is a little deep thought, so there's going to be some questions in your notes that are going to be in part 2, which are going to be a little cerebral. So make sure you put your thinking cap on, okay? And bear with me as we go through. So these words, again, you've learned these words in biology class. Most of you, the greater majority of you. So diffusion is passive. No extra energy is put in from the cell or from the system so that particles or molecules can move from high to low concentration. It's all about nature wanting to have a balance. All right? Facilitated diffusion is, again, no extra energy, but using those channel proteins we learned about in the previous set of notes. And then osmosis. Osmosis is, for some reason, confusing for some people, but let's try to clarify that, okay? Water is the key here. Osmosis is the movement of water from higher to lower water potential, and that is the symbol, that little trident is the symbol for water potential. That's why you see that in your notes, and you will see it on the next slide or 2. So we'll talk more about that. So if you look at this diagram here, this semi-permeable membrane, which some of you have under your sinks in your kitchens, you see you have the salt molecules are red, everything else is blue, water is blue, and you see how there's a difference in concentration, high concentration to low concentration. Now if you don't put any energy in whatsoever, you're not forcing water in one direction or another, over time, the movement of the solvent from low to high solute concentration, well, in this case, it's the water, you're moving the water is going to move from here, which is the solvent, the universal solvent, water, is going to move from the lower to the higher. Okay? From the lower to the higher, which is going to make the water levels change, but balance as well as it can that concentration. That's the goal, okay? And we're going to look at what water potential is more in a minute. That's why it could be a little confusing. Okay. So understand the concept of water potential, that symbol, and explain how dissolved solutes affect the water potential of a solution or a cell. Because that's what all this comes down to. Yeah, we've got beakers and pipes and diagrams, but eventually we're going to be talking about how, in the next set of notes, I believe, next set, how this affects fish and invertebrates like coral polyps, okay? All right, so water potential. What is water potential? Potential energy, right? PE, of a solution compared to pure water. Water will move by osmosis, we said that already, from higher to lower water potential. So here's the key. That asterisk is telling you what this means. The more water molecules in a solution, the higher the water potential. So if you have a higher concentration of solutes in one container or one side, this is going to be a higher, this one's going to be a higher water potential than that one, okay? Because the more water molecules in a solution, the higher water potential. So here, pure water, solutes. The moment you add solutes, you decrease that water potential number. Pure water is going to have a Agar. Why is this not working? Previous, thank you, okay. Sorry. Pure water is going to have a value of 0. All right. So if a solute is dissolved in water, the proportion of water molecules in the solution decreases the water potential. That's what I just said. So these are keys to understanding that difference. And different solutions, if we're talking about an estuary where you've got fresh water coming in, if you're talking about the mouth of a river or the open ocean, you're going to, compared to where that fish or organism came from, you're going to have different types of solutions that have different water potentials compared to the other ones. Okay? So hypertonic, isotonic, and hypotonic are also words that you should have learned in biology class, or maybe even in chemistry class. Okay? So hyper, greater, like hyperactive, you have a greater activity. So the solution has a greater concentration of solutes. It's then the organism. We're talking about the organism. So a lower water potential than the cells and body fluids of the organism. Isotonic, same, right in the middle. So it's typically where an organism would be happiest, right? Okay? Same concentration. Water potential is the same as the cells in the organism, okay? And then hypo, lower, opposite of hyper. If hyper is greater, hypo is lower. Some of you athletes may know about hyperextending, or extending greater than it's supposed to go, or hypoextending. So lower solute concentration. So a higher water potential than the cells, but that's the confusing part. It's got a lower concentration, a higher water potential, a higher concentration, a lower water potential. Get those cards going for Unit 6 and word them correctly so that you remember this stuff, these differences. Because remember, you have to explain yourself with the differences here. And then apply this to a question that may say something like, a fish swims from the ocean to an estuary or the mouth of a river. In terms of hypertonicity, or water potential, describe the reaction that the cells are going to have, and so on and so forth. That's the kind of crazy question you could have. And you have to understand these concepts and remember them at that time, right? Okay, so diffusion and osmosis using plant tissue and non-living materials. So we are going to, now I'm going to present to you three, I think, scenarios that will be discussed probably in the future, but you have to just understand them. And they're really simple, okay? They're actually really simple. It's just maybe stuff you haven't seen before or heard before. So, this is Visking tubing. It's also called dialysis tubing because it is made out of cellulose material that is semi-permeable, selectively permeable membrane, like a cell's membrane. Whoa. So that's why it's used for demonstrations like this. Now we are going to, tomorrow, listen to a woman go through the process of using Visking tubing in a demonstration, okay? A demonstration about glucose and starch. Glucose is a monomer of a long starch chain, right? So imagine trying to get a 12-inch ruler in your backpack versus the meter stick at the front of the classroom, you know? One of them is not going to fit. And so that's what Visking tubing is representing in the cell. The glucose is small, it will go through, but the starch is too big, it will not go through. The starch molecule is too big. And that's what that demonstration is going to show. And that's essentially what this demonstration is showing here, okay? So you have this solution in the Visking tubing, and you drop it into water. The solvent is just water. And then over time, you're going to see that the smaller particles diffuse through the Visking tubing, but the larger ones do not. It's actually quite simple and not necessary to do a whole demonstration in the classroom. That's why I'm doing it in visualization here, okay? So hopefully you filled in this material in your notes. And so this is essentially describing what's happening in these pictures, okay? So if I may describe these three different scenarios, this is an order, and then this is just, nothing's gonna happen because it's just pure water inside and out, okay? That's kind of a control. So this is got the sucrose solution in it now, and then it's going, the particles, because sucrose is just a glucose and a fructose. So it's still kind of small. And sucrose is the white sugar you cook with or bake with, or put in your tea or your iced tea or your coffee. And so over time, that sucrose solution is small enough to go through the Visking tubing and into the surrounding water, and then leaving the water behind, so the water level drops. That's all. It's rather simple, okay? That's Visking tubing, or AKA otherwise known as dialysis tubing. Now, we've got a lot here about calculating surface area to volume ratios. In order to do that, we have to have some basic information, and this looks a lot like math, but you really can't have science without math, can you? So, this really requires no explanation. All you have to do is in your notes, get down the formulae for each of the surface areas for these different shapes and the volumes for these different shapes, okay? Three-dimensional shapes. So cuboids and spheres and cylinders and cubes. Now we're going to talk more about what this means soon, okay? What these numbers mean. These are just here kind of for reference for the shapes we're going to be talking about. And I only included the cylinder because it is mentioned in an activity that you're going to do, or a lesson you're going to do in the workbook next week, okay, as a way to remember all this stuff. So, but we don't, there's no spherical cells, I don't think. Probably somewhere close, similar sphere, in a plant or something. Not spherical. Cylindrical. Sorry, was I saying spherical the whole time? Cylindrical. So make sure you're getting these right. These formulae are just simple plugins. Plug in the numbers. We're not really going to need these for the notes after this, but we are going to need those in that activity next week, okay? So you should be getting close to finishing. 10 more seconds. All right, so let's discuss what some of these pictures mean, because you're going to have to answer some stuff in your notes about them. So, surface area to volume ratio. You can see what's going on here. This is just a visualization of cell growth, perhaps. And this is a description of why a multicellular existence would be better than a single cell existence, okay? And so the point here is the larger the surface area to volume ratio, the harder for materials to diffuse in. And I'm going to show you how we can see this. And it makes sense, right? I mean, the bigger something is, the harder it is to get something from the outside farther inside. You could consider this classroom. Let's say we had to move all the furniture into a brand new classroom or any room, right? And the bigger the room, like think about a factory or a warehouse floor, you're gonna have to move the furniture you have to expend more energy. You have to move the furniture farther into the building than if the room was this classroom size. You'd only have to move the furniture in a little bit. So it's really a simple concept. And so the smaller the surface area to volume ratio, the easier it is for materials to diffuse from the outside to the inside, okay? And then surface area to volume ratio is, if it's too small, you're going to decrease the rate of chemical change. So like I said, because of this, the rate of chemical exchange is going to go down. Those materials needed. But if you multiply your cells, then you have a smaller surface area, and so it'll be easier to get materials from outside into those individual cells than it would to be one big cell. And this also is the reason why we don't have ants and bees the size of your table walking around in the world. It has to do with this, surface area to volume ratio. All right, this section of the notes is interesting because it lets you visualize the movement of a material, the diffusion of a material from the outside to the inside. And it's using something called Agar, A-G-A-R. Some people say Agar. That sounds like someone's name. So Agar is probably the best way to say it. And these are cubes of agar that have essentially a food coloring in them. Let's just call it that. And then they're dipped into an acidic solution. In this case, it's just vinegar. Vinegar is acetic acid. And over time, the acid neutralizes the coloring until you get to, the end of your experiment. And then you can take a ruler and you can measure how far the new volume or the new surface area is, and then determine what the volume is based upon the surface area, right? And you can see, you can compare the size to the change. So you can see a much more diffusion towards the center, and then this one's completely, the color's gone in that one. So, and then this is your, these are your measurements. So cube side 1, 2, and 3 centimeters. The surface area of each is measured here. The volume is 1 cubic centimeter, 8 cubic centimeters, and 27 cubic centimeters. So what you're going to see, so the volume is increasing at a greater factor than the surface area. So this can all be determined before you actually do this experiment, okay? Because all you're doing is measuring these things. But this is an 8-fold increase. This is only a 4-fold increase. And then a 2-fold increase, 3-fold increase. So again, the volume is increasing at a greater factor than the surface area. The surface area to, there should not be a hyphen there at surface area. I don't think. The surface area to volume ratio actually goes down. The larger cube, this ratio decreases. And so, and then this is a visualization, an actualization of this determination, okay? And what does that mean? Take a look at this graph. Study it for a moment, and then you're going to write a generalized conclusion to this graph. You can see that you've got, on the y-axis, you've got surface area to volume going up, and then on the bottom, you've got the length of a side going from 1 to 13 in centimeters, yeah, okay? So what generalization, if as this does this, this does that. You think you got it? As the cell size, because we're talking about cell, which is analogous to a cube, as the cell size increases, the surface area to volume ratio decreases continually. I mean, it's more drastic when you're in this end and it kind of levels out, but it's still going down. So the conclusion here, the conclusion here is anything that comes into a cell, like oxygen or food or nutrients or, or goes out of it, like any waste products or even materials for other parts of the body, proteins and stuff that it made, must travel across the cell membrane. We know that. That's obvious. But as the cell grows larger, like the cubes, 1, 2, and 3 centimeters, right? The ratio of surface area to volume decreases dramatically. Dramatically, right here. This would be considered a dramatic decrease. Just like those Agar cubes. I almost said Agar. Larger cells must still transport those materials across their membranes. But they have a larger volume to supply and a proportionately smaller surface area through which to do so. So this is the conclusion we can take from that demonstration. All right, we're moving on. So you're going to have to revisit those notes and study that concept. Same with this one. This one's, I think, actually maybe a little simpler. This one is requiring us to understand the effects of taking a plant tissue. In this case, these are pieces of potato, little core samples, I guess, of potato. And they're all the same length, and they're put into solutions of sugar, 4 of them, and then a control with distilled water. That's the experiment. Simple. And you're going to, so that's different water potentials. So remember, this is pure water, so it's going to have 0, and then it's going to decrease slightly, go down. The water potential is going to go down as you go in this direction. And then you're going to leave them in there for a specified amount of time, and then you're going to remove them, dry the water off, and then measure the if there was a length change. So essentially the swelling of the tissues. So in order to estimate the water potential of those tissues. So the analysis is here. The percentage change in mass for each potato cylinder is calculated. So before and after, and then you divide, and then you multiply by 100. So we've done this before, percentage change. You subtract, and then you divide, and then you multiply. I'll show you that on the next slide, okay? So if there is a potato cylinder that has neither increased nor decreased, there's no overall net movement. That's really simple. That's because the solution of this particular cylinder was in the same water potential as the potato itself, as the cells of the potato itself. So there was no concentration gradient and therefore no net movement of water into or out of, that's supposed to say out of the cells. Remember, if there's any questions or you don't understand something that's happening here, raise your hand and that guy over there will pause the video. All right, so continuing that same experiment, the potato water potential experiment, you can see the concentration of sucrose, inside the potato cylinders can be found if a graph is drawn showing the percent change. So you take the data, so you've got your masses. We weren't supposed to measure the length, supposed to measure the masses. I don't know what that length thing was for. Anyway, so see how the concentration of sucrose goes up. And then so therefore the water potential would be going down here. The initial masses are all relatively the same. And you can see the final mass, you got a decrease, kind of the same here, little mild decrease, quite an increase, and then a huge increase on that one, okay? So the change in the mass is here, and the percentage is here. I told you, it's subtraction, and then a division, and then a multiplication. So the final mass, for example, 5.8 for the distilled water one, minus the initial mass is going to give you an answer, which is your numerator, and then your initial mass denominator, you divide those two, you multiply by 100, and you get your answer, okay? So then with that, you plot a graph. Change in mass percentage, that's here, by the concentration of sucrose. And in this case, they put 0 in the center with negative at the bottom and positive at the top. And these are their plot points. X's, not dots, remember? Okay. And that's a line of best fit, right? So it's a pretty simple demonstration that just tells you what you're doing is you're measuring the water potential, I went back, of the material, the plant material that's in here. And you can do this for any plant. You could take a carrot, and then you could do this with cells as well. You might not use sugar. You might use something else. I don't know, but you can also put tissue samples from animals and do this the same way to see what the water potential is for to estimate water potential of those tissues. That was the purpose of this. Okay, and we can see them here. If it's the same as the original, the water potential was equal. All right. Moving on. Explain the movement of water between cells and solutions with different water potentials, and explain the different effects of plant and animal cells. So we've got hypertonic, isotonic, and hypotonic solutions. So these are plant cells on the top and animal cells, these are blood cells actually, on the bottom. So hyper is greater than, so the water is going to move out to the area that has higher solution, higher concentration. Isotonic, you're going to have an even flow, and you can see the cell wall protects the shape of the cell, but you're still going to have a decrease of that vacuole. And then here the vacuole is going to swell. With the hypotonic, that means water is going to go into the plant cell and swell. So what would happen to a fish? This is in your notes, and because you can apply this to any plant or animal, it's the same effect is going to happen. You're going to get swelling and bursting, possibly if it's animal. Plants don't usually burst, or lice, it's called lysing. But here you see this bursting cell. It's just too much water going in there. This is a nice environment, and here the cells shrivel up. So if you were to put a fish into a hypotonic solution, like a saltwater fish into fresh water, what would happen to those fish's cells? And what would the water look like? It wouldn't be pretty, would it? That fish's gills would start to lyse, and the water would turn bloody, and it would be very sad. So that's what you have to know. You have to know about how these different water potentials affect living organisms, okay? So you were just supposed to label these in your notes, that's all, and describe what happens. So you're labeling the different scenarios, and then with a little description that describing what's happening in each of those, okay? Plant and animal cell. Okay, not far now. So here we're talking about, I'm going to go deeper into these individual organisms in the next set of notes. But for now, this is just an introduction to respiration, moving waste products and raw materials in and out from the surfaces of organisms. Yes, the gills are a surface. They're protected by an operculum. They're protected by that covering in a bony fish. Operculum, gill cover, see right here? But it's still exposed to the outside. I mean, you could kind of consider the lining of our lungs, even though they're inside, they're touching the air, right, from the outside. So surface of an organism. And these are, which we'll talk about later, these are analogs. Gills are analogs of mammalian lungs. So these are generalizations. We're going to get into the specifics soon, but you can see that even through the tissue, the tentacles, and the more tentacles they have, the more surface area they have, and so the more exchange that can happen through the epidermis. They also exchange with each other. These are coral polyps, right? So the cenosarc, remember we learned about that tissue that connects different polyps? They can exchange through that as well. And then this is, lungs, so here's our lungs. These are the little grapes called alveoli in our lungs, where it touches capillaries, and so the carbon dioxide diffuses out and we breathe it out, and the oxygen comes in and diffuses in and travels through the circulatory system that way. Well, the same thing happens in fish. And we're going to go deeper into this, but the water passes through the mouth, flows over the gills, and if you zoom in, you've got the lamella, which you already drew in that one workbook exercise. That's increasing the surface area, and the capillaries run adjacent to and in those lamellae, which exchange the gases. It's very similar. It's an analog to mammalian lungs, our gills. They do the same thing. They're just structured differently. Same structure. Sorry, same purpose, different structure. Analogous. All right, surface area to volume is dependent on size and shape of an organism, and relate this to the need for specialized gaseous exchange surfaces and transport systems in larger organisms. Okay, so just kind of fill in the blanks here. This is a really good picture. We're going to talk more about what these pieces are and how they relate to what we're talking about. This is not a dead fish. This is not a cut open fish. This is just holding a fish so that you can pull the gill cover up, the operculum, and expose the gills. And this is where they breathe. This fish would hopefully return back to the water again. But smaller organisms have a larger surface area to volume ratio, and their rate of diffusion is faster than larger organisms. We've already determined that with the Agar demonstration, okay? So larger organisms, like us, we need gills or lungs to be able to increase that surface area. And then we have to transport it around our body. So we need arteries and veins and capillaries. A single-celled organism or a very small organism like an ant doesn't need that. They can just diffuse all the materials through. Increased surface area increases the area through which gases can diffuse. That's a simple concept. And then be transported. It's like the tentacles I showed you in the last slide with the coral polyps. The more tentacles, the more gases can diffuse. The more gill area, the more gases can diffuse. The more alveoli in our lungs, that's why we have two lungs, and it's easier to breathe than with just one lung, and they're branched out like this, increasing surface area. So gills, here's the pieces of the gills that I was referring to before. Gill arches are these cartilaginous, I believe they're cartilaginous, I don't believe they're bony, arches. And you see them back here again. These are the gill arches, okay? They're arched. And they hold the filaments. The filaments are the pieces the red stuff. The filaments have lamellae. Remember, so they're broken up into even smaller fanned out pieces. And the lamellae increase the gas exchange surface area. And then these things are called gill rakers. And so this is that folded like that. Yeah, so here are the rakers. You're going to label this in your notes, so start labeling. The filament with lamellae coming out, the capillaries, the incoming with oxygen into the rest of the body, and outgoing with carbon dioxide, so oxygenated and deoxygenated. Know that I'm actually blue, and so people think we have blue veins or animals have blue veins. It's not, it's just in diagrams it shows blue as deoxygenated. It's just a little darker red. It's like a maroon maybe. Okay, venous blood versus arterial blood, but it's definitely not blue. And this is another, just a close-up view. It's the same concept. All right. One more slide after this and we're done. This is just a way, so if you were to look at the lamellae of a gill and they looked abnormal, like this, or, you know, broken apart or squished, that's called a gill hyperplasia, and that's caused most commonly by parasites or bacteria infection, or even poor water quality will damage these tissues. It shortens them, rounds them, fuses them even together, increases mucus production, and reduces the surface area for gas exchange. That's what gill hyperplasia does. And we actually learned about this word when we learned about parasitic relationships in organisms in the first semester. We learned about gill hyperplasia. And then our last slide today is what does the flow of blood in a typical fish look like? The circulatory system of a typical fish. So it's very similar to a mammal, except if you were to look at a mammal, this would be the heart and these would be the lungs, rather than the gills coming down both sides of the fish. So blood passes through the muscles and other body tissues and releases oxygen into those tissues and picks up the waste product carbon dioxide. That blood, that venous blood, is pumped back to the heart, then to the gills to pick up more oxygen and drop off the carbon dioxide, and then that blood leaves the gills through arteries and goes back and the cycle starts over again. And like I said before, gills are analogs to mammalian lungs. And that finishes up our notes. You do not have to know the parts of a heart, so that's just there for showing you the difference between perhaps a mammalian heart or like a human heart and a typical fish's heart. It's a little bit different shape, isn't it? If you're familiar with a human heart shape.